Good afternoon, ladies and gentlemen. On behalf of the Department of Pathology, I welcome you to the second annual Professor Barrie Hanchard Memorial Lecture, which marks the start of the CPC calendar for the 2020-2021 academic year. I believe that Professor Hanchard would have been pleased to see the lovely turnout, as CPC was very dear to him. And this evening, he would have been especially proud that his son, Dr. Neil Hanchard, is presenting in this forum. Dr. Hanchard's lecture twists and turns to learn and discern a diagnostic genome sequencing case is quite intriguing in title, and I am sure will provide an exciting and educational evening. I am, as I'm sure you all are, looking forward to his presentation. And so without further ado, I hand you over to the moderator for this evening, Dr. Mahiri Bromfield. Dr. Bromfield?
Thank you, Dr. Gibson. Welcome, everyone. And I'm just here to be the interim. So next, I'll ask Dr. Shirley, a senior lecturer in the Department of Pathology, to begin our program with a word of prayer.
Good evening, everyone. And I invite us to just take a minute as we settle in, just quiet ourselves for a moment, and I invite us to pause for this moment of prayer. Let us pray. Gracious God, we give you thanks. We are not where we were a year ago, but we are dispersed now across many miles, more conscious than ever of ties that truly bind, of that which truly endures, good values and attitudes, and sound medical practice. We give thanks for Professor Hanchard's life and contributions, in particular to our specialty of pathology. And we pray that, encouraged by his example and that of so many great UWI stalwarts, we may press on in these challenging times and hold to the tenets of robust medical practice. We are sure that Prof is smiling this afternoon as we have gathered to listen to his son. We are grateful that we are sure to hear echoes in a very special way of him this day. And may all go well with these proceedings. We give you thanks, O God. Amen.
Our Dean, Dr. Tomlin Paul, Dean of the Faculty of Medical Sciences here at the University of the West Indies Mona campus, will now bring greetings.
Thank you very much, Madam Moderator, Dr. Bromfield, Dr. Agri-Irons, our distinguished colleague and friend, Dr. Tracy Gibson, Head of Pathology, Dr. Carl Bruce, our Medical Chief of Staff, our distinguished guest speaker, Dr. Neil Hanchard, members of the Hanchard family, members of the staff of the Department of Pathology, which is, I would say, the extended family of Prof. Hanchard, and our even wider extended family, members of the Faculty of Medical Sciences, students, staff, colleagues, or special guests. It is a pleasure to bring greetings on behalf of the Faculty of Medical Sciences, Mona, on the second annual Barrie Hanchard Memorial Lecture. Universities have a tradition of hosting memorial lectures, often named in honor of a former faculty member. And these lectures bring outstanding individuals and scientists to our campus and provide special opportunities for students and faculty to interact with speakers. As we engage in this evening's memorial lecture, we have the double pleasure of celebrating the memory of a distinguished son of our faculty, Professor Barrie Hanchard, and at the same time, to hear from the son of this distinguished son, Dr. Neil Hanchard, who was a graduate of our medical faculty also in 1999. So there seems to be some genetic connection here. And I noticed the title of the talk, talking about genome sequencing, twist and turn. So let's see if we get the secret coming out here. But the event this evening is more than hearing our distinguished speaker. Celebrating Barrie Hanchard's life and indeed his dedication to the Faculty of Medical Sciences is an opportunity to reflect on our history as a faculty. That part of our history that remains with us and never dies, but we propose that it is our duty. I propose, in fact, that history is our duty to keep it growing. And some people call that our legacy. The Greeks have a rich legacy of literature. Jamaica has a rich cultural legacy. The Faculty of Medical Sciences has a strong and rich legacy of excellence. Excellence in teaching and training, excellence in research, excellence in service. And Barrie Hanchard's time with us demonstrated that excellence. He has made a tremendous deposit to our legacy bank. As we reflect today, I challenge you to use the legacy of Professor Barrie Hanchard to embolden us, even at this time during the pandemic, when we are stretched and uncertainty and all of that, and beyond this period. What would Prof had said during this time is a reasonable question to ask. What would Prof Hanchard have said to us in that quiet tone, that quiet but deep wisdom? And we can answer that question, in fact, because the legacy of Barrie Hanchard has been left with us in our hearts and minds and spirits for all of us who knew him, who worked with him, who were taught by him. We have had that touch of his legacy living on with us. And so today is really a special time for us to have this memorial lecture in his honor. And we will never forget this special man that was in our midst. Let me close by thanking all who have worked to plan this evening's event. Special thanks to our head of department, Dr. Tracy Gibson, and a special thanks to our distinguished guest speaker, Dr. Neil Hanchard, and all of you who have come together for us to celebrate this moment. I do wish for you all and for all of us a special time of connection and intellectual nourishment as we hear from our distinguished speaker. Thank you very much, Madam Chair.
Thank you, Dr. Tomlin Paul. I now ask Dr. Carl Bruce, Medical Chief of Staff from the University Hospital of the West Indies, to bring greetings from that institution.
Thank you very much, Chair. Dr. Sonia Thomas, Dr. Neil Hanchard, Dean of the Faculty of Medical Sciences, Dr. Tomlin Paul, Dr. Agri Irons, Dr. Tracy Gibson, the Head of Pathology, fellow speakers, UHII and UII faculty, ladies and gentlemen, students. It's with high honor that I take the opportunity this evening to, on behalf of the University Hospital, offer these few words at this second Professor Barrie Hanchard Memorial Lecture. On behalf of the Chairman of the University Hospital of the West Indies, Professor Evan Dugan, and the Board of Management, our executive, our students, and staff, we feel that it's a privilege for the Hanchard family to allow us to participate in the second annual lecture. The hospital, as many of you know, is currently fully engaged in the COVID-19 global pandemic. But one thing that many of us have noticed and would have noticed is that many of the leadership of the various departments in the hospital would have gone down to room 14 during this pandemic and would have been seeking the advice of Professor Hanchard. I would say that, generally speaking, that most departments, if not all, have been performing commendably throughout this global pandemic since we diagnosed our first case on the 10th of March, 2020. And I feel that there can be little doubt that the skills that Professor Hanchard has given to many of us is what is guiding us, despite the fact that we are not able to go down and to gain, as Dean says, from his wisdom. We can take comfort, and I hope the family of Professor Hanchard can take comfort in what we will do for our country and what we will do for our region over the next year, year and a half, while we continue to battle this public health crisis. But I also want to say that it's been a privilege knowing him as friend, teacher, and mentor. And as Dean Paul said, as we share in his legacy and as we listen this evening to the lecture that his son, Dr. Neil Hanchard, himself a scholar, will deliver, we cannot help but think about the rich history of the University Hospital of the West Indies since it opened in 1952. I feel that the knowledge that has been shared with us from Professor Hanchard and many others is what will take us through as a university hospital for the next 70 years. So I would say this evening, Chair, that while we are here for the lecture, we celebrate the man, we celebrate his teachings, we celebrate his friendship, we celebrate his dress, we celebrate his sports, we celebrate his cars, we celebrate with his family, we celebrate his knowledge of food, we celebrate his watches. It's been a privilege to have been taught, as many of my colleagues have been. And we thank you very much for allowing us to participate. And we look forward to a great lecture this evening. Thank you very much.
Thank you so much, Dr. Bruce. Dr. Jacqueline Jagan, Senior Lecturer in the Department of Pathology, will now present a biography of Professor Barrie Hanchard.
Thank you, Dr. Bromfield. Good evening, all. Professor Barrie Hanchard was a graduate of the University College of the West Indies in the medical class of 1967. He pursued his residency training in pathology at the University of Toronto and subsequently joined the staff of the Department of Pathology at the University of the West Indies in 1976. During the course of his distinguished career, teaching and research were important parts of his life. He was best known as the principal investigator for the National Institutes of Health-funded Human T-cell lymphotropic virus type 1, HTLV1 project, which produced landmark discoveries, important public health initiatives, and several local and international accolades, including the UWI Vice Chancellor's Award for Excellence in 1999-2000. He was a founder of the Doctor of Medicine DM program in pathology at the UWI, which serves the Caribbean region, and head of the Department of Pathology from 1996 to 2004. Until the time of his death in August 2019, he was a director of the Jamaica Cancer Registry and the chairman of the weekly CPC conferences of the Faculty of Medical Sciences, UWI-MONA. Prof, as he was affectionately called by most, was the consummate professional, meticulous, accurate, and dedicated to his work in practice, teaching, and research. For this, he was honored by the government of Jamaica in 2011 with the Order of Distinction at the rank of Commander. He valued history, especially that of the Faculty of Medical Sciences, UWI, as well as lifelong learning, but had many other passions, including fashion, fast cars, fine wine, photography, and sports. Prof. Hanchard was always available, supportive, and genuinely interested in everyone with whom he worked, mentoring many medical students, residents, pathologists, and others beyond the medical fraternity. He was very proud of his family and spoke of them a lot, often sharing stories and photographs. Professor Barrie Hanchard is greatly missed.
Thank you, Dr. Jagan. Before we have the introduction to our guest speaker, I'd just like to indicate to you, the audience, that we have two formats by which you can communicate. We will ask you to put your questions, if you have any, about the main presentation in the question and answer section that you should see at the bottom of your interface, and any comments or greetings in the chat. So we will look specifically at the Q&A section of the webinar at the end of Dr. Hanchard's presentation. We'll have a 10-minute spot for addressing your questions and hopefully answering most of them. So at this time, I invite Dr. Agri Irons, consultant psychiatrist, to introduce our guest speaker.
You have been suitably greeted by the Dean and the Chief Medical Officer. But I want to send a special shout out to our friends who have joined us all the way from Canada, in addition to the friends and family who are here to represent. Today, the second annual Barrie Hanchard Memorial Lecture represents the fulfillment of a promise made one short year ago. Today, we have the distinct pleasure of being addressed by Neil Andrew Hanchard, the one and only son of our distinguished and beloved professor. I am sure that this will not be the last time that the names Mendel, Watson, Crick, and Hanchard are mentioned in the same sentence. Because Neil Andrew is not only a pediatrician, but also a medical geneticist of international fame, he may well be seen to have two distinct professional pathways. Much more than that, he's also a speaker and a teacher with an impressive list of publications that would be the envy of any professor in any academic institution anywhere in the world. Neil Andrew represents the Hanchard brand, and in the words of his beloved father, he is a gentleman and a scholar. Neil's tertiary education began at our own University of the West Indies, where he greeted the new millennium as one of those rare honors graduates of the Bachelor of Medicine and Bachelor of Surgery. His strength of character and all-round excellence combined to earn him the Rhodes Scholarship, which allowed him to express himself as a rare Doctor of Philosophy, D.Phil, at the University of Oxford. Further studies have earned him the distinction of being a fellow of the American College of medical genetics. He is currently the youngest member of the board of the American College of Genetics. This combination makes him not once, twice, but 3 times a doctor, a rare combination indeed anywhere in the world. Neil Hanchard's journey began with his own special genealogy. His 2 grandfathers were well-known teachers and educational administrators. His 2 grandmothers were well-known teachers. His mother, Dr. Sonia Thomas, a consultant pediatrician and administrator, a great teacher in her own right, and his father, whom we revere today, professor of pathology, donated the Y chromosome. All of these persons provided the genetic template for his present activities. Dr. Neil Hanchard was successful not just because he was born that way. He also acquired his erudition and urbanity from his Jamaican home training, not homeschooling, but home training. Neil excelled as a national representative in water polo and swimming, and as a more than competent schoolboy footballer at Wolmer's. He is also an accomplished pianist. He has assured us of further genetic continuity by having 2 sons with his charming wife, Dr. Susanna Hanchard, née Legister. The Hanchard brand continues. From his base at Baylor University in Texas, Neil continues his work as a team leader in genetic research, unraveling the mysteries of pediatric disorders and other rare disorders, including, of course, sickle cell disease, with a special focus on diverse populations, which has led him through the mighty continent of Africa, from Egypt in the north all the way down to South Africa. In addition to his international reputation and individual brilliance, Neil is recognized by his fellow scientists as an exemplary team member who facilitates the growth and development of his fellow team members. Sounds a lot like the professor, doesn't it? Neil, the pediatrician, supplements his vast research activity with continued clinical consultations regarding rare and sometimes exotic childhood disorders, which pleases his mother no end. I have no doubt that Neil's genesis and genetics will positively affect the future of mankind. To paraphrase the words once spoken at the River Jordan, this is our beloved Son, in whom we are well pleased. Listen to him and learn. Neil, it's over to you, son.
Thank you, Dr. Irons. And now I invite Dr. Neil Hanchard to present his talk, Twists and Turns to Learn and Discern a Diagnostic Genome Sequencing Case.
Okay, so hopefully everyone can see me. And I'm going to try and share my screen. So hopefully everyone sees that as well. So first, thank you very much for the organizers for inviting me to come and give this talk, but also for remembering the legacy of my father. He really believed in CPC. He felt that for students, this was the way to link pathology with the clinics. And this was really something near and dear to his heart. And he would have been very happy to know that this is how we're honoring him. I'd also like to thank all the speakers before. Hopefully all protocols have been observed. And a special thank you to Dr. Irons, whom I know as Uncle Agri, and who my kids know as Uncle Grandpa, not just for those very kind words of introduction, but also for his many years of support and for setting the bar last year, the first annual talk, so high that I'm sure that I'm not going to be able to make it, but we're going to give it a go. So the title of my talk today involves twists and turns, which is a double entendre of sorts. And the first twist and turns refers to DNA. And DNA, of course, is the sort of building blocks of life. And it is packaged, twists and turns, into 23 chromosomes, 23 pairs of chromosomes in each cell. And the sequence of DNA is made up of the 4 nucleotides that you see there. And at specific points in the DNA, these code for amino acids. Those amino acids, when aggregated, then form a protein. That protein then leaves the cell and goes to do its functions. And this is very much, this central dogma of going from a gene to a protein is very much the core of medical genetics, where the attempt is to look at genetic changes and link those to specific diseases. And it turns out that my affinity for medical genetics is in many ways hereditary, but I did not know about this beforehand. And I only found out about it when I was invited to be a guest speaker at the valedictory service for the greatest boys' school in this hemisphere. And as a memento for that, I was given a small keepsake. It's a small wooden box you see here. And inside that box was a book, a small book, which is labeled The ABC of Medical Genetics. And this book, as you can see here, was from 1969. And so the table of contents reflects what we knew about medical genetics in 1969. And this book came to be with Wolmer's because it had been gifted by none other than my father, whose signature you see there. And this was many years before I even knew anything about it. So it's a full circle. So what I hope to do today was to give you a flavor of the medical genetics in my DNA by talking a little bit about genome sequencing and how that is becoming the standard for diagnosing rare disorders in medical genetics. And then talk a little bit about a local case which illustrates the twists, the other aspect of twists and turns, which involves a diagnostic odyssey which required many different minds here at UWI to finally solve. And my hope is that by the end of the talk, although we're taught in medical school to think horses and not zebras, we will find that sometimes what you're looking at is in fact a zebra. So to start, I thought that the best context to give for where we are now in DNA sequencing is to start back with the Human Genome Project. And this was a multinational, highly ambitious project at the time to delineate the sequence of the 34 billion nucleotides that make up the human DNA or human genome. The first draft of this was made public in 2001. And there are two major things that came with this first draft that have really function to catapult DNA sequencing. The first was that it provided a reference map for genetic variation. So now we knew how many genes we had, we knew where those genes were, we knew the sequence of those genes, we knew what was in between those genes. And that allowed us to do mapping of genetic variation to diseases a lot faster and a lot easier than we had previously. The second thing is that it paved the way for genome sequencing to become cheaper. And that's what you see illustrated here. So the first human genome, in the Human Genome Project, the first efforts cost $100 million per genome, and that is not a typo. But now, in 2020, the cost of a human genome is hovering around $1,000 US. And that is an astronomical drop in cost that is beyond even what you'd see in computing, which is the white line here, where Moore's law talks about the fall in software, sort of computer chips, which is what allows your cell phone to have as much power as your desktop did 20 years ago. So this is really a massive drop in cost generated through the Human Genome Sequencing Project. So in the course of the genome sequencing project, we then realized that actually the vast majority of your genome is not coding for any specific protein. In fact, there's only a small proportion, maybe 2%, that actually codes for a protein. And that 2% is sort of now colloquially referred to as an exome. And the exome is, because it's only 1 to 2%, it's easier to sequence the exome because you don't have to do all the genome sequencing, and it's also cheaper to do. And so was born the advent of exome sequencing, which allowed us to basically interrogate all the coding sequences for the known disease genes in one go. And one of the first efforts to sort of demonstrate this had to do with a syndrome known as Kabuki Syndrome. And Kabuki Syndrome is a very rare neurodevelopmental disorder. It occurs in maybe 1 in 40,000 to 1 in 80,000 individuals. And it includes a number of different features. Some are congenital, such as congenital heart defects. They can have skeletal problems. But one of the most striking clinical characteristics are the facial features, and you can see those here. So you see these very long eyelids, and they often have long eyelashes, so very long palpebral fissures. And in many ways, sometimes the eye is actually even everted, which you see a little bit here. And so Kabuki syndrome has been around since the 80s. It was described in Japan and given its name because of the similarity of the facial features to the Kabuki mask, which is used in Japanese theater. And although clinically, this was a syndrome that we knew and could describe, we didn't know exactly what the genetic defect was that caused this. And so researchers from the University of Washington took it upon themselves to do exome sequencing in a group of about 10 such individuals. And when they did this, they identified that all of these individuals, most of these individuals, had changes in a gene called MLL2. And unfortunately, genes have terrible names, so we just have to live with that, but it's called MLL2. And what they found in MLL2 is that the changes that were in the DNA there led to a change in amino acid, were very rare because this is a rare disorder, so they're not seen in the general population, and they were very damaging to the resulting protein and its ability to do its function. And so for today, if you're following along at home, especially for the students, those are the 3 things that I want you to remember. So the DNA changes the amino acid, is very rare in the population, and it causes damage to the resulting protein activity. So after these initial descriptions, many groups, including our group there at Baylor College of Medicine, started to publish their own experience with exome sequencing. And here you can see the results from the first 250 individuals. And I'll say that this is 250 individuals who have, they've had lots of testing, they've done lots of genetic workup without an answer. And if you just look in the red box, you can see that we're able to find a diagnosis in about 25%. So that's 25% on top of whatever was there before. And in fact, if you look at neurological disorders, almost a third of these disorders could be explained and diagnosed using this modality. A couple of years later, sort of repeated this on a much larger group. So now we're up to 2,000 individuals. Again, we're seeing that same 25% can be solved. But because we had more individuals, we could actually look across the age spectrum. And what was interesting is that for a fetus, so that is where you have an ultrasound and you can actually see that there are features in the fetus that are suggestive of the genetic diagnosis. You could almost find the answer in 50%. So this was a huge boon, and so became sort of the soup du jour for genetics going forward. And that's what's sort of reflected here. So, you know, around the time when that book that my father had given to Wolmer's was being made, this was all about syndromes and describing these syndromes in a clinical fashion. And then with the Human Genome Project, and our ability to do this kind of exome sequencing, you can see that the number of gene discoveries for these rare Mendelian conditions, for these rare syndromes, went up astronomically. And therefore, the number of new syndromes that were being described also went up astronomically. And the only reason that these two things don't match completely is that there are many genes that can be found for one syndrome. So Kabuki syndrome, for instance, has 3 or 4 different genes that can cause it. And so this kind of exome sequencing, which is represented in the blue bars here, that's certainly become almost the go-to for genetic testing in current medical genetics. And it's such that with this sort of expansion of new diagnoses and new syndromes that we're able to describe, if you went back and looked at that group of 250 where we had like a 25% diagnostic rate, that level is almost up to 50% just from the new genes and the new syndromes that had been identified. And if you look at that cohort that we had 2,000 and we're around 25%, from those new genes again, just in a matter of 5 years, we could get our diagnostic rate up to 40%. And that's on top of the rest of the genetic diagnoses that we can do. So this was a real boon. And so you might be thinking, well, these are kind of rare disorders. These are 1 in a million, 1 in 50,000. You know, is this really something that we need to know anything about? And you'd be right to one extent, except that there are like 7,000 of these very rare disorders. About half of them occur in children, and about more than 80% of them are known to be caused or thought to be caused with good evidence by faulty genes. And so that means there are somewhere estimated to be around 300 million people worldwide who have this kind of rare disease. And I'll also go further to say that outside of North America, Europe, and East Asia, we really don't know exactly how many people there are because it's just not been looked at just yet. So what's also really good to know as a medical geneticist is that we're moving now into having treatments that are available for a lot of these disorders. So once you know the cause, it's a lot easier to have a treatment that goes with it. And so, but treatment aside, there are many different reasons to try and find a diagnosis for these kind of rare disorders. One would be to end the diagnostic odyssey. So again, you know, most of these families, they've been to 3, 4 doctors. You know, it's been 10 years of trying to look for an answer. And so being able to have a diagnosis at the end is so comforting to so many of these families. It's also a case with many of these syndromes that they may have heart problems in future life, or they may have kidney problems. And so getting that diagnosis allows you to do medical screening so that you can have early diagnosis and early intervention as needs be. But it's also important for the family. So understanding and knowing what's the likelihood of this happening again, or, you know, what should I tell my pregnant sister? So there are many different ways in which a diagnosis is really helpful in this group of disorders. And then, as I said, the really exciting part is that now, once you know what the actual cause is and you have a diagnosis, now you can start to transition that into treatment of disorders. And there is a very good example that's been published about this, and it was in a set of fraternal twins. They're well known, they're known as the Barrie twins, and they were, shortly after birth, found to have just lots of problems. So these were really colicky babies, they were throwing up, they had significant delays in their development, they were sort of low tone and were described as floppy. And then when they began to be 5 years old, it got even worse, because now they're starting to lose their skills and regress in their development. And that regression also included some very abnormal hand and foot movements that was ultimately diagnosed as dystonia. And the pediatric neurologists in the room will know that the most common cause for dystonia in a child is a deficiency of dopamine. And in those kids, if you give L-Dopa, you can make a dramatic improvement. And this was certainly the case for these children. The provision of L-Dopa significantly improved their well-being and quality of life, but didn't stop anything. So when they were seen in our institution at age 14, they still had problems with drooling, particularly at night. They had vomiting, there was unsteadiness, and they had problems with breathing on occasion. And so they were sent for doing genome sequencing, and genome sequencing revealed, and again, so our big three, our golden trifecta for today, these are rare amino acid changing, protein damaging variants in a recessive configuration in this SPR gene. And SPR, it turns out, is important in making a cofactor. And that cofactor is important for the production of not just dopamine, for which you're giving the L-Dopa, but also serotonin. And so in truth, these kids are deficient not just in dopamine, but also in serotonin. And so it was recommended that their L-Dopa should be supplemented with a serotonin precursor, and that's 5-HTP. And that worked wonders. So here are the twins just before they went to college, and they started to have improvements in the coughing and breathing problems. They had more stability. And in the paper, they describe handwriting changes going from before treatment to after treatment. And they had handwriting experts come and attest to how amazing an improvement this was. So this is a good example of kind of where diagnostic sequencing can take you to sort of management and treatment that can make a big difference to a family. So now we want to sort of translate that more into a local experience. And this was a clinical case that was referred to me by my father. So I was home for holidays with my family one Christmas. And as is often the case in our house, there was a discussion in the back veranda. And in the course of that discussion, my father confided that, you know, a young lady had approached him and had asked him for help and asked him to come to me and speak to me about genetic testing, that she had seen a number of doctors and they wanted to do genetic testing, but it wasn't readily available, and asked if I could help. And true to my father's form, he, on the back veranda, sat down and asked me very carefully if I would help. And I can remember him saying, you should just give her a call. Just give her a call, right? And so said, so done. I gave her a call, and that started down a long diagnostic odyssey, which I'm going to relate to you today. So I had the occasion to see this young lady and her son in TMRU in the office of Professor Colin McKenzie, God rest his soul. And at the time he was 2 years old, and he had been seen by his pediatrician at 6 months, had some small developmental concerns, and was so sent to a pediatric neurologist, whom you see there on the lower right side, at 9 months of age. And his history was such that he had a pregnancy and delivery at 36 weeks, pretty unremarkable, appropriate for gestational age. There was a little bit of neonatal jaundice early, but it was highly responsive to phototherapy, and no other abnormalities were found. Developmentally, prior to that 6-month period, he wasn't too bad. He had started to get some head support. He was sitting with a little bit of support at 5 months and was starting to reach for objects. And on his examination at that time, the most marked thing was that he was on the small side, so his weight was at the 5th centile, and he had this very marked truncal hypotonia and unsteadiness. And you can see a little bit of that in this video here. It's not, it's a little bit, it was taken when he was a little bit older, but hopefully you'll still get the point. So you can see that as he's trying to crawl, there's a lot of unsteadiness. You can see the titubation in the head and the unsteadiness in the body. So our story continues. He was then referred for physiotherapy and for further investigation, which we'll come to. And then at 11 months, he was noted to have this kind of horizontal nystagmus and was referred to ophthalmology. And by the time he was seen at 18 months, he was noted to only be babbling with about 3 words in total. He could understand simple one-step ungestured commands. He could play peekaboo, so he had good cognitive skills. And although his nystagmus had abated, he now had these roving eye saccades. And you can see that kind of illustrated here. So as he is trying to track, if you look carefully, you can see that the eyes start to bob up and down, back and forth as he moves across. And at the time, he was still having significant truncal ataxia and hypotonia. So even though he could sit unsupported, he could only do so for a relatively short time. And you can see some of that illustrated in his gait and walking here. You can see a wide-based, high-stepping gait. You can see the swaying of the body from one side to the other and the difficulty in being able to have tone. But he was otherwise healthy. So this is the point where, you know, my father would typically say, let's hear from the students. So for the students in the audience, what do you think? Or maybe it's easier to say, you know, what more information do you want? What would you like to see? And so I'm going to ask you to put your answers in the chat. And if you don't, then I'm just going to start calling people from the names that I have here. So what else would you like to see? What else would you want to have done? Great. So I see MRI brain. I see a CT to rule out a brain pathology. There's another call for an MRI. There are more vociferous calls for an MRI. Okay. So we're going to fast forward the story, and it's not possible to get through a talk by a medical geneticist without including a pedigree. So here is the family history. And so squares here represent males, circles represent females, and the shading represents affected individuals. So here really, although we have many people in both sides of the family, there's really only one person who has these kinds of features. So for the students, again, send me a chat and let me know, like, if you see this, what is the most likely mode of inheritance? What should you be thinking about in a pedigree like this? Okay. I see X-linked recessive, I see recessive, I see autosomal recessive, I see X-linked, autosomal recessive. Yes, recessive. Should be your first thought. There's also the possibility of what's called de novo dominant. So that is it's a dominant disorder, but you're the first person, sometimes the only person in the family who gets it, and that's de novo dominant. So, but if you see it on exam, right recessive, and if the dominant doesn't have de novo, forget about it. Okay, great. So now we're going to start to get some of the investigations that people are talking about. So he had an EEG after he had a spell. That EEG was relatively normal. He saw ophthalmology who said that he did have some visual impairment, but the major issue was of these cogwheel saccades that we described before. And he had a hearing evaluation, which showed bilateral sensorineural hearing loss. So we can add that to our differential. And then to please everyone else, he had an MRI of his brain. And the MRI showed that he had a 2 by 2 by 2 centimeter arachnoid cyst in the temporal lobe, which even I could find on his MRI. So he went and saw a very prominent neurosurgeon in Jamaica who happens to be with us today, and he felt that there's no evidence of a mass effect, no other abnormalities, arachnoid cysts are common, and this is unlikely to be the cause of the clinical presentation. And the rest of the MRI was otherwise normal. So other people asked about various other kinds of testing. Other people asked about other kinds of testing. So he had normal electrolytes, normal liver function tests, a serum lactate. Yeah, there's a metabolic screen, which we commonly do to look for very common metabolic disorders. So plasma amino acids, urine organic acids, and acyl-carnitine profile. He had a full workup, and all of these, some of which you see over here, all of these are normal. So someone said we should do a karyotype. And it's worth pointing out that now we've sort of moved from karyotypes, not completely, but to do what's called a chromosomal microarray, or a CMA. So karyotypes, which you can see illustrated here, you sort of look at the chromosomes under the microscope, and you can see large, you can see certainly missing chromosomes, which is the case here. But you can also see where there might be large missing pieces or extra pieces or chromosomes that are stuck together. But nowadays, we're sort of moving towards doing a chromosome microarray, which is able to look for both these big changes, but also small extra or missing pieces of DNA, especially if they occur over a gene. And like a karyotype, it does all the chromosomes in one go, but we found that diagnostically, you go from a sort of 5% yield of doing a karyotype to a 15% yield in doing a chromosome microarray from a diagnostic standpoint. And this is what that kind of looks like. So the black dots here at the 0 line represent having 2 copies, which humans do. And then you can see these regions here where you have a pronounced block with a number of, with a loss, suggesting there's only 1 copy available. And this is a deletion of this particular region. And of course you could have an increase, which is a gain or usually a duplication of that particular region. So we were able to get a chromosome microarray done, and this revealed nothing. Stone cold normal. So at this point, someone says, well, you know, could this be something else? Could it be something environmental? Could it be, you know, how do we know it's something genetic? Which we don't. But as I said, rare disorders, sometimes it's a zebra. And so we felt that we shouldn't stop here, that this was an opportunity to do diagnostic sequencing. And this was back in 2013 when diagnostic sequencing was not nearly as cheap as it is now. And so we felt that there was a need to get some sponsorship. And in order to get sponsorship, there was a need to write a begging letter. And so we wrote a begging letter to the Ministry of Health, outlining the case for doing exome sequencing in this individual, and making a strong argument as to why it was important. And we got the reply from them, and lo and behold, they said yes. So we were able, again, with the help of the folks in TMRU, to take a sample, send this off to our genetic lab at Baylor, and we did whole exome sequencing, we held our breath, and we got a result. And this result was implicating a gene called ACO2, or aconitase, in our case. But sometimes these things take a little bit of interpretation. So we can take what we've done before and sort of walk through what we're dealing with here. So the first thing, of course, is that this inheritance pattern, or AR, is autosomal recessive. So again, recessive makes good sense, and is consistent with the pedigree that we have. And you can see that 1 variant is inherited from the father, 1 variant is inherited from the mother. And then we have our trifecta again, okay? So does this lead to a change in amino acid? Yes, it does. Both of them, you can see there's a change from proline to lysine here, and a change from arginine to cysteine here. So check. Is this rare? It's a rare disorder, so it should be fairly rare. So you can again see that in databases which are only about 10,000 individuals at that time who had this kind of sequencing. There's only one individual with this change, and no, this variant had never been seen before. So again, rare, check again. And so then we say, well, does this cause some damage to the protein? And although we don't have direct evidence, we certainly have computer programs that can make an assessment as to how likely this is to damage the activity of the protein. And the two computer programs you see here both agreed that this is a kind of change that's bad, right? So check. So we have some strong suggestion here that these changes are causing, could be causing a problem. And so here they are represented in the ACO2 gene. Again, humans are diploid, we have 2 copies of everything. So there's a change from dad on one and a change from mom on the other. And this fits what's called a compound heterozygous recessive model. And actually, this is by far the more common way in which recessive disorders present. So we tend to think about like sickle cell, where you're homozygous for one change as being the prototypical recessive, but this way of getting what's called compound heterozygous is actually way more common in rare disorders. So what do we know about this aconitase and ACO2? Have we ever seen anything like this in medical genetics? And at the time, there were only 5 cases, 5 individual cases reported in the literature. But interestingly, those 5 cases had a fair degree of overlap with the features that in our patient. So there was this kinds of eye phenomenon. There was this kind of ataxia and hypotonia suggesting of a cerebellar abnormality, although in our patient, the MRI was normal at the time. There's another patient who had hearing loss. So we've got some genetic evidence. We have a couple other examples that suggest that maybe this is indeed the right diagnosis. And so what is this ACO2? And one of the nice things about medical genetics is that you get to tour everything. So, you know, now you have to go back into your biochemistry. And so if aconitase sounds familiar to people, it's because it's the enzyme in the Krebs cycle that converts isocitrate into citrate in the mitochondria. And for the students, enzyme deficiencies tend to be recessive. And this is because enzymes have a lot of redundancy. So if you look at this graph here, if you have 50% enzyme activity, which is kind of like being a carrier for this disorder, you still have almost 90% efficacy. You can still do the job. And it's really only once you get below that 50% that you start to see the efficacy of the enzyme dropping off. And so this is why, if you see it on your exam about a metabolic disorder, the most likely answer is, again, going to be recessive. Great. So I mentioned mitochondria, and the mitochondria are individual organelles. There are multiple copies that are found in each cell. The mitochondria is important to the generation of energy through the production of ATP. And one of the areas where there's a heavy preponderance of mitochondria is in the brain, which is why you can get some of the features that we saw in our patient. And although their mitochondria each have their own DNA, and so you can have primary mitochondrial disorders, in a large proportion, most of the proteins that are used by the mitochondria are actually encoded in the nucleus. So what you find is a lot of these sort of secondary mitochondrial disorders where it's an abnormality of something that just gets used by the mitochondria. Okay, so at this point, we've come pretty far. We've gotten ourselves a genetic looking diagnosis. We see there are a couple other cases that sound very familiar, and the computer programs are telling us that this is going to cause a problem. But computer programs are sometimes wrong. So we wanted to be able to test and see whether or not these variants really did cause a problem. And to do this, we had some help from Debbie Thompson in TMRU, who performed a punch skin biopsy on our patient. She sent us a little flask of that. We were able then to culture fibroblasts from the skin, and then we can use those cells to perform specific assays to look at function. So that's what we did. So the first thing we did was we looked at the activity of the enzyme, right? So we have some normal fibroblasts from a controlled individual, and that becomes sort of full normal activity. And you can see that in the cells from our patient, we're looking at about 20% activity, and that's a highly significant drop. So below that 50% threshold. And actually, Nowadays, in the cellular systems, we can actually put back in that gene. So that gene, we can essentially rescue the defect that you have in that gene in the cells. And when you do that, you see that you can completely rescue the phenotype back to a normal level of activity. So it's important to realize here that the amount of protein is actually normal. So here's the patients and here are the controls. Normal levels of protein. It's just that the protein itself is not doing what it's supposed to do. And there are ways to kind of evaluate how well the protein is acting, and that's a cellular respiration assay. So I'm just going to walk you through this. So on this axis is oxygen consumption, and that, just think of it like the cell is able to kind of breathe and do respiration. And so if you start over here on the left, this is basically basal respiratory activity of the cell. And you can see that the control in the blue and the patient in red, not that much of a difference. Then you give this, the cell this oligomycin, and that then causes some of the mitochondrial effects to shut down so that you're entirely dependent upon ATP. And so now you can look at just ATP dependent respiration. And again, our patient and our controls look about the same. Then if you give another drug, which basically like frees up everything, and now the cell is operating at maximal activity. So this is like, we got to run 100 meters. We got to have maximal activity. And now you get to see that there is this difference where our patient isn't really able to maximize their oxidative capacity. And that's primarily the big difference that we're seeing. And conversely, in the rescue cells, we're able to get that back up to normal. And then you give another compound to get back to baseline. Okay. So after all being said and done, so now we have our clinical features for our patients alongside the other patients that have been described. And what you can see is that when you're at sort of 20 to 30% activity, generally these individuals are going to present in that first year of life. They're going to present around somewhere between 5 or 6 months with the features that we saw in our Chacha boy patient over here. Conversely, if you have very little activity, so here 5% or almost zero activity, these individuals are going to present very, very early at birth. And these are the individuals who end up in the NICU. They are sick babies and require a lot of support. Okay, so here we felt like we'd gone through this diagnostic odyssey. We felt like we had a fairly confident genetic diagnosis for this young man, where he carries both abnormal copies, and then both parents are carriers. But I did say that this is useful information. And that's because about a year later, mom gave me a call and said, hey, guess what? I'm pregnant, and I'd like to know if my new baby girl to be is going to have the same problems. And so we were able to work with Dr. Nadine Johnson to be able to do an amniocentesis. The cells were sent to Baylor, and we don't have to do exome sequencing. We can just look for those changes that we're really interested in. And we could do that in for the amniocytes and the fetal cells. And when we did that, she turns out to be a carrier. So expected to be just like her parents, and I can attest that she certainly is. And then, history tends to repeat itself. So mom called back again about a year or two later, was pregnant again with another girl. And sure enough, we were able to do the same thing and show that she had another carrier girl who is also doing quite well. Okay. So hopefully by the end of all of this, end of all of our twists and turns today, I've convinced you a little bit about the utility of diagnostic genome sequencing. This is fast becoming the standard of care for rare disorders and in the field of medical genetics, because it's efficient, it's becoming increasingly cost effective. There is some training and expertise that's required, but it affects almost every system and almost every medical specialty that there is. And so, this is my sort of shameless plug to say, if you have cases where you have this kind of suspicion, let me know, drop me an email. And for the students, if this is something that you found interesting and want to pursue further, especially that expertise and training, again, drop me an email. So that is all that's left to me to do is to just give my largest and heartfelt thank you to the Department of Pathology. This was absolutely my father's second home. There were times where it was his first home as well. And a special shout out to Dr. Bishop and Dr. Gibson, who were also very close and very special to Daddy, as well as Dr. Bromfield you see here. And just to thank you for the opportunity to continue his legacy in this way. And with that, I'm happy to take questions.
Thank you so much, Dr. Neil Hanchard, for a clear, informative, and very interesting talk that brings us up to date on the modern applications of exome sequencing and how it can really change the way practice, our current practice, and influence treatment, that it's not just something in the textbook anymore. You do have a number of questions in the Q&A, so I'll remind the audience to please put your questions in the Q&A. The first question is from Sian Gilman, and she's asking, is exome sequencing the only method of genetic testing, or is it the easiest and most cost-effective?
Yeah, so Sian, that's a great question. No, exome sequencing was sort of the, something that could be done to sort of look at all the genetic code in an efficient way before. Nowadays, we're moving towards doing more of just sequencing the whole genome because the costs have come down so much more. And the advantage of doing the whole genome is that you can get a twofer. So you can look for missing pieces and extra pieces of DNA, but also be able to look for the sequence changes that are occurring and their interpretation.
Okay, I hope that answers your question, Sian. You also had another question. Is it possible to tell when a condition is recessive or de novo dominant?
Once you get the sequencing back, that usually is really where the answer is. I think de novo dominant is something that we've learned is much more common than we thought. So these used to be the disorders that people would just say, oh, it's a sporadic type of thing. But it's not always, it's not easy to tell if it's at all possible to tell before you have the testing. There are some disorders that congenital defects tend not to be recessive, but some are. So there are general classes that can help you, but for the most part, you gotta wait to get back the sequencing result.
Okay, thank you. There was a question which I think maybe applied to when you were talking about the workup. Somebody asked about metabolic screening and toxic screens.
Yeah, I think toxic screening is a good question. I don't think we've done a lot of toxic screening. My default is that by the time they get to me, then somebody wants to know if they have a genetic disorder. So I am trying to look at a genetic disorder, and it's not clear that some of the toxic and environmental screens sort of are clear one-to-one. So, you can get an abnormal tox screen, but whether or not that's actually causing, for environmental pollutants or things like that, whether or not that's actually causing the disease is hard to tell. So again, we sort of tend to work on the DNA side because again, we can easily interpret those.
Okay, thank you. There's another question from Cherie Mayer. What is the cost for whole exome sequencing? I think you had mentioned that, but I think...
Yeah, so I mean, back at the time, exome sequencing, was sort of somewhere between $3,000 and $5,000 US, but now that's come down a tremendous lot. Now, it's hard to do a one-to-one comparison because there's like insurance and all this kind of stuff. But, we can do exome sequencing on a research basis for hundreds of US dollars now.
Okay, that's definitely relatively cheaper. Someone else has another question, Sian again. Were you able to treat the truncal hypotonia?
Yeah, so I feel like this is one of those really unfortunate ones where we would love to be able to offer an easy treatment. So, we can do these kinds of gene rescues in the cellular context. And, we asked around and we looked at, doing bone marrow transplants and stem cell transplants to try and identify something that might be able to sort of fix the defect that we could identify, but we, to this point, really haven't. So this is one of those ones where, you know, the diagnosis we have, but the therapy is going to take seeing more patients and being able to see what can be done. Great question.
I think someone has a related question. They say, Dr. Hanchard, why were you not able to perform the rescue technique you described?
Yeah, so I mean, we did do the rescue, but we did it in a cellular context. And there it's sort of artificial. And certainly I think that people are thinking about how you can do that kind of change, you know, maybe in something like stem cells and then give them back to the patient. And I think that some of the ethical considerations, but then also some of the sort of consequences that may not be immediately obvious are something that the field is moving towards to be able to do, but it's not quite there yet. But that's an excellent question. That's where we want to get to.
Thank you. Another question from Warren Blake. What about its use in genetic counseling in people about to have children?
Yeah, so I think that this also encapsulates a lot of the other questions that I saw, which is sort of how we utilize this kind of data in a prenatal setting. And I think that there is, for instance, carrier screening, which you basically can do sequencing of a lot of these genes to determine if one or both parents have these kinds of changes that would make them a carrier for a recessive disorder. And that kind of carrier screening can occur, you know, preconception, and is something that is being done very widely to be able to then talk about counseling about what your chances of this happening. Someone else asked a related question about whether or not you could do this using non-invasive prenatal screening. And the answer is nowadays, yes. You know, at the time we were sort of limited, but nowadays limited to just looking at large aneuploidies. That's, you know, trisomies and that kind of thing with that kind of technology. But nowadays we're able to do much more sequencing from cell-free fetal DNA. And so nowadays that's something that would certainly be able to be done in the prenatal setting. So that's an excellent question. And I know someone else, someone else asked about the harmony, and I think it would be all these things fall into that kind of category.
There's some related questions in terms of Dr. Jamie Lee Foote is asking, since we know that meaningful mutations can occur outside exons, how does whole exome sequencing compare to whole genome sequencing? How much would we miss with whole exome?
Yeah, so that's a really clever question. So, you know, the exome is what we can interpret. So we actually, you know, we have a good idea about the central dogma, and you change the sequence, you change the amino acid, you change the protein. And the problem outside of the exome for the sequence data is that it's not really clear how to interpret it. So, you know, somebody else asked about what does the other 98% do? And it turns out that that 98% is really important in sort of regulating how these genes work and when they're turned on and how they're turned on and which cells they're turned on. But we don't have a very clear sort of central dogma once you get outside of the coding region. And so some people feel like the coverage that you get with genomes is better for the coding region, like you don't miss anything. Whereas depending on where you are, you can get exomes that are going to cover that coding region just fine. But I do think, as I said, we're moving towards whole genome because we can start to look for missing and extra pieces of the DNA in addition to the coding variation. And we have all these big projects where we're starting to learn about exactly how the genome is regulated. And so we might be in a position going forward to interpret changes that are occurring outside of the coding region. So that's an awesome question.
Thank you so much, because someone else had a similar question. They were asking if the exome is only 2%, what does the other 98% code for? So I think you've addressed that one. Someone is raising the point, Dr. Mike Mills, will we be prepared for possible genetic discrimination? All genes matter?
That's right. I'm going to get a shirt with that on it, Mike. Thank you so much. Yes, I think that genetic discrimination is a real thing. I mean, in the United States, they have the GINA law, which is a President Bush initiative that basically didn't allow for any discrimination on the basis of a genetic diagnosis, so that you couldn't have your insurance, you couldn't be denied insurance for a genetic diagnosis and that kind of thing. But there are loopholes, and some of the aspects of that GINA are falling apart, and we're starting to see the consequences of that. So it's a real thing, but I think that that's going to be something that has to be addressed in the sort of legislature and advocacy side of things, to put in place, right, the appropriate securities beforehand. But it's a great point.
Dr. Warren Blake had the similar point that the role, how does it influence insurance acceptability and patients being denied insurance? So
Yeah.
you've addressed that. People are asking for your email address so they can contact you.
Sure.
And they're asking how the child is doing presently.
Yeah, so I think Mom is on the call, and he's doing well. He is still needing physiotherapy. He's in school. He's in a mainstream school. He gets special. He has a tutor who helps him. He's been relatively healthy, but he still has, as is often the case with sort of his kind of developmental delays, he's still somewhat behind from a chronological standpoint, and so needs a lot of help otherwise.
Okay, thank you. There's a question.
Chair, I do have a question.
Sorry.
I think their hands up as well.
Okay. All right. Yes, there are quite a few hands up. Sorry. I'm trying to keep track of all the questions by using the Q&A. Someone has raised the point about genetic testing being accessible in the Caribbean, or will it likely remain within the domain of academia? And if we're talking about zebras, that this may be hard to sell to Caribbean nations.
Yeah, so I think that that is, so I actually have the opposite view. And some of that view comes because I've now worked closely with the guys in Africa. So in Democratic Republic of Congo, in Cameroon, in Nigeria, in Uganda, they now have facilities to do genome sequencing. It's the same thing that you do when you sequence the COVID genome. You're just doing it on a much larger scale. So it's certainly possible to do. As I said, the trick is actually in the interpretation. That's where the hard part is. And I also think that there's a lot of stuff that masquerades as other things that is actually a genetic diagnosis. And the truth is that we just don't know. So again, the case that I presented here, may never have had an actual diagnosis of a genetic disorder. And I suspect there's lots of this that occurs, throughout the Caribbean. Certainly in Puerto Rico, for instance, they do genetic testing. They have a lot of founder mutations for very specific genetic disorders, and there's no real reason to think that that doesn't occur here as well. I just think that we're not looking for it. And my hope was that in doing this, we could sort of stimulate the idea that we can do more of this. But there is. I've been addressed, and people have emailed me about this kind of thing previously.
Sorry. There's some raised hands. Dr. Agri-Irons has a question.
Yes, indeed. Neil, your lecture was a thing of beauty. And I guess that means it's going to be a joy forever. Thank you very much. You mentioned the closeness in production of dopamine and serotonin on one particular gene. What is the future for diagnosis and treatment of, and in fact, forecasting of depression based on what you're seeing with serotonin and dopamine?
Yeah, so that's a trickier question, because I think the bulk of evidence suggests that unlike these very rare Mendelian disorders where you can link, you got a problem in this gene, you get this disorder, depression and schizophrenia, a lot of these more common diseases are a little bit more complex. So you're talking about multiple genes that interact. You're talking about genes that may interact with the environment and specific stressors and epigenetics and all kinds of other things. So I think it's been frustrating when I speak to the psychiatrist friends, because they feel like they haven't been able to they haven't been able to do a kind of direct diagnostic switch in going from sort of seeing a patient with this to being able to do the sequencing. But I know for a fact that there are people who are trying to create sort of composite scores that might talk about your risk for depression or your risk for schizophrenia and other neurodevelopmental psychoses, with and then talking about doing genetic testing for treatment-related issues and people who are treatment-resistant, for instance. And so there might be an avenue there where I think it is, and this is definitely something that people are working on.
You did mention in your rescue situation, I believe, that you gave a serotonin precursor and it had good results. I mean, we've been trying to help serotonin along by various means. Is there a future for precursors in the treatment of depression?
Yeah, that's hard. I think the problem is that, again, so this is very clear because you have a very specific, these kids had a very specific defect that caused a, not a small change in the serotonin, right? This is a big deficiency. And so you could see it being utilized somewhere in there. I suspect that as we start to get a better handle on the composites of these psychiatric disorders, we might be in a position where you could make specific recommendations about how to attack a specific pathway, or if using sort of combinations of drugs might allow you to address a particular pathway in terms of treatment. And part of that cocktail may actually be some of these precursors to either move things up or down. But I would, if I were a betting man, which I am not, I would bet against the idea that there's going to be a kind of magic shot in that regard.
Okay, I'll wait until Christmas on the back veranda and we'll have further discussions then.
Absolutely.
Hope you'll be able to make it.
Absolutely. Well, that's the big question. Yes. I look forward to it when I can.
Thank you. Dr. Bruce, you had a question?
Yes. Thank you very much, Chair. So Neil, congratulations on a great CPC. I think the fact that you have attracted over 450 participants this evening meant we would have had lecture theater. But back to your lecture, Neil, if I could just take you a little bit off, and that would be then into the question of precision medicine. And with the genetic testing, even if the tests are not unique, you take us down a path where you have to do precision medicine to treat some of the conditions that you can now diagnose at hitherto no treatment. But that comes at a significant cost. So if I put on my chief of staff hat, how then are countries such as ours, developing nations, are going to be able to afford where we're going to go in terms of modern treatment for some of these disorders that you're going to diagnose, if the drug companies are going to find specific biomarkers and now develop precision medicine to treat these conditions?
Yeah, that's a tough question. I think it's the same question that's being struggled with again in various parts of Africa where we've had the chance to have this conversation. And there isn't an easy answer. In most cases, it takes, there has to be a kind of partnership. So that's kind of the model that most people are going with. So it's usually a public-private kind of partnership or a government-private type of partnership to allow this. And often also partnerships that may be outside of the Caribbean, so maybe partnerships with the drug companies to try and leverage, very unique genomes, very unique clinical scenarios to be able to get some of those costs down. I think naturally as these things become more common, some of the costs will come down, particularly for the sequencing. But there's always this sort of haves and have not kind of argument, which should be difficult. But I do think it's the kind of thing that people are more aware of now, and there's a lot of upfront negotiation to try and ensure that there's not the kind of sort of colonial academics that used to happen previously. It's an awesome question.
Thank you very much, Neil.
Thank you, Dr. Hanchard. I'm going to take one more question from the floor in terms of hands up. Dr. Indira Singh Mainat, you've had your hand up for a while, I think. You can go ahead and talk. You can unmute. Dr. Singh Minor, do you still have a question?
Thank you, Neil. It was really wonderful. It's been a long time. I think most of the questions have been answered, actually. It was an excellent lecture. I do miss academia. I have several children here in Angola. It's a very small population, and because of the very small population, not necessarily inbreeding, but we have the genetics is obviously very close, and I have several children that don't have diagnoses with developmental delay. I think I've made an attempt to get genetic testing on some of them, and I didn't end up with any positive results. So they're just there with developmental delay and in a special educational, special needs. And so I guess I'm looking forward to probably, in terms of research, how we could get these kids enrolled in the future.
Yeah, no, I think that's it. And that's kind of goes to the original question. I am sure that these cases are knocking about there. These are not, it is highly unlikely that there are no, there's not a substantial genetic burden in the Caribbean. And the trick will be, I think the first thing you have to do is you have to sort of get some work in partnership. So there are a number of programs now that will do sequencing for free for diagnostic purposes, but you kind of have to be in the know. So, send me an email or a Facebook message. And I think once we start to see it, then I think we're at a position where now we can sort of engage governments, universities, et cetera, that, these are problems that are occurring in your population. And so there is some, need for governments to think about that as part of the broader, health perspective, where it really hasn't been up to this point in time, but you kind of have to prove it first, which is the hard part, I know.
Correct. Because as usual, I would find certain families that I found that there were seizures in certain families and certain parts of the island. Then I found out they were cousins. And I just found it very interesting that I could probably, I knew I knew the history. And then I found out that some of the children or the siblings never made it past infancy. And then it turned out that the mother and the father were cousins, and so then it went back several generations.
Yeah, I think that I think as Dr. Warford put in the question and answer area that I think ultimately the goal is not necessarily to have a government be a part of this, but I think that the Caribbean is going to have to work. I think there's a potential for the Caribbean to work as a group to sort of centralize testing and be able to have that be sort of the way. So you can sort of take your cases, we can say, oh, this is what you need to send, and that sequencing can be done somewhere in the Caribbean. But I don't think we want to have genetic testing labs everywhere, but I think we need to have that facility be available and not sort of every physician who has an interesting case kind of like wandering around trying to figure out how to get testing done.
Okay, thank you, Dr. St. Minot. I hope that addresses your question. Dr. Hanchard, there are a number of people with a lot more questions. Would you be willing to accept questions by email? And
my inbox will hold them, sure.
If your box will hold them. And there are a couple more that, as you said, you've seen the discussion in the chat about the regional approach to addressing the need for testing. Someone is asking a question about testing, whether a geneticist, how do you view the challenge of differentiating between what is possible and what is permissible?
Yeah, I think that's a hard question that the whole field is kind of struggling with. But I think that there is kind of a consensus that is developing that sort of differentiates between where, you know, everybody kind of knows that's not okay. You know, that ethically, there are a lot more consensus. There's a lot more consensus statements about what is ethically reasonable, and how, and in particular, when things comes to sort of germline genome editing, people, there's been enough bad things that have happened previously that people are very circumspect about how to approach that going forward. And so I do think that there's as a field, you know, this idea about ethics has to go alongside. And I think it also, like, the ethics that are appropriate in one setting is not necessarily true in another cultural setting. And so I think we have to define those things for ourselves in you know, in particular areas and groups.
Thank you. There are a couple more questions. I think you've addressed some of the group questions. I think Loren Small is asking, what's the trick to being good at genetics? You spoke about the tons of really randomly named genes.
Yeah, so I would not say I'm good at genetics yet, but I'm getting there. You know, I'm fortunate enough that, you know, I'm basically surrounded by human genetics all the time. But even I have gotten to the point where, you know, I can't tell you which syndrome is caused by which gene. So I'm pretty good with the common ones, the ones that we see commonly, but honestly, you gotta look stuff up. And being able to be okay with the resources and being able to find, because some of this stuff, you know, was published like last month or the month before. And so keeping up with it is very difficult. And so I think people have given up on the idea of remembering gene names at this point in time, except for the really common ones. So don't feel bad. There's no sort of magic to it.
And Siobhan is asking if you could just recap the rescue technique. She wasn't quite understanding it.
Yeah, so it's kind of harder to explain. It's kind of a gene trap where basically, like, because you know where the gene is and you know the sequence of the gene, you can, in a cellular system, actually just, you can buy these genes off the shelf and you can put the coding DNA back in using, usually I think it's like a lentiviral kind of system that will allow you to basically just insert it back, usually beside where it is, and then it can sort of express itself.
Okay, thank you so much. I'm afraid I'll have to stop the questions at this point, but from the general feedback in the Q&A, some people are suggesting that whether you'd like to spearhead a regional laboratory, as there appears to be a great deal of interest and need for it. So
absolutely, and I'm gonna presume that that also comes with the requisite resources and finances to set that up. But I'm happy to do it and happy to put my name to it.
And someone asked, how much does the lab cost?
Yeah, I think those are all very real things, and it goes back to the idea that I think that having, you know, a lab in each country doesn't necessarily work very well, but I do think that there are opportunities to do sequencing in a more regional sense, and that can be, the sequencing machines are not as expensive as you'd think. That actually the expensive part is sort of maintaining it. And then being able to get the reagents in and be able to like get maintenance done on them and those kinds of things. Those are where the big problems are. But, you know, it's comparable to a hematology lab or, you know, a chemical pathology lab.
All right, so thank you so much again, Dr. Hanchard. I think everybody has really enjoyed your talk and it has stimulated a lot of questions and thoughts. So thank you so much. And at this time, I'm going to ask Dr. Karen Bishop to give the closing remarks.
Good evening, family, friends, colleagues. I think this was a fabulous evening. And you know, to comprehend that the reason we're gathered here we're really happy that we could do it again for a second year, and especially since Prof's son could be our very special guest. I don't think we could ask for it to get better, despite why we're here. And that we could do it despite COVID. So COVID has really thrown us wide this year, and despite all the challenges of COVID, we're here doing this. And while COVID has stopped us gathering in person, the little blessing is that doing it virtually has allowed so much more of us to join. So we had 646 people registered. And as Dr. Bruce mentioned, just over 450 of us joined in live this evening. I don't think we could, I think it's a fabulous way to honor Prof. Hanchard. So firstly, we want to thank Dr. Thomas, Prof's wife, and of course you, Dr. Neil Hanchard, for your blessing for this event. And we're really happy that your family and close friends could join us today from Jamaica and beyond. Dr. Neil Hanchard, your dad always spoke of you with such great pride, and I know he would have been very, very proud today. Thank you so much for your beautifully delivered lecture, for the story, for patiently addressing all the questions and the comments, and for making this very complex subject very clear for us. I think everyone remembers this is a gift that your dad was blessed with, and you have certainly inherited it. Dr. Agri Irons, we really appreciate your insight into Dr. Neil Hanchard, the professional, but also into Neil, the multi-talented son and dad and husband. Thank you so very much. Dr. Tomlin Paul, Dean, Faculty of Medical Sciences, UWI-Mona, and Dr. Carl Bruce, Medical Chief of Staff, UHWI. We know that your schedules are very, very hectic, and so we really appreciate you taking time to help us honor Prof. Hanchard. And we thank you for your very insightful and sometimes personal and genuine words. Dr. Tracy Gibson, our department head of pathology and chairman of the CPC committee. And I smile when I say this because Prof did this on his own for more than 30 years. I don't even know how many years, but now it takes a little committee of us to do the same thing. So Dr. Gibson heads this committee, and she worked tirelessly to make sure everything was organized and perfect for today. And she made the life of her little elves very easy. She continues to guide us in the department with a steady hand and with a calm spirit that I know would have made Prof very proud. Mr. Damian Brown, head of the faculty's IT team, assisted by Ms. Shanique Graham. And Damian is literally responsible for making this virtual event possible. He worked very hard to make it happen. And because of he didn't just do that, because of his great respect for Prof. Hanchard, he insisted on directing this evening's proceedings personally. We can't thank you enough, Damian. The beautiful invitation and program was designed by Dr. Sophia Green. Dr. Sophia Green is a new pathologist. She literally joined our staff on Monday, but we were expecting her for a few weeks. And so she came in the nick of time because we are not necessarily talented that way. So thanks a million, Sophia. Thanks to all the other members of staff who helped to take us through the evening. Dr. Mahiri Brumfield for eloquently guiding us through the program. Dr. Suzanne Shirley for the opening prayer. Dr. Jacqueline Jagan for sharing Prof's biography. We also had support from Mrs. Karen Black Grant. She normally sends out all messages for CPC. Ms. Hyper Nelson and Mrs. Lorraine Grant Davis in our office. Thank you all so much. We look forward to doing this again next year and pray that it will be possible. The last thing I have to say is that this evening marks the beginning of the CPC calendar for the academic year. For now, it will be virtual. So we hope we can continue to have lots of you join us throughout the year. And as they say in Jamaica, walk good. And nowadays we have to add, be safe. Thank you, guys. Thank you all.
Bye-bye. Thank you again.
Bye, everyone. Have a good evening.
Thank you.
Bye all. Take care. All the best.
Thank you again, everybody. I really appreciate it.
Thanks, Dr. Neil. Great, great talk today. He would have been very proud.
Thank you.
Yes, ditto. Magnificent, Neil. Really, really good.
Okay, thank you. Thanks, Neil. Take care.
Thank you. Take care.
Good.
Bye. All right, be safe now, everyone, and thanks a lot. Take care. Bye-bye. Be safe.
Bye.
Well, thanks.
Bye.
Everybody.